the Q&A feature, and we will circle back to those after Dr. Bertrami's talk.
Okay, I thank Stephanie and Davor for their very nice introduction. I'm really honored to be here today. I'll share my screen right away, and we'll start with the presentation. So, first of all, I would like to thank the Journal of Molecular and Cellular Cardiology for choosing this as a paper of the month. I was really thrilled about this, and I would like to thank also the International Society for Heart Research for giving us the opportunity to present our data. As soon as we came back from the United States, as previously introduced, we continued focusing on the heart as well as on other organs. And in this first paper that came out from our lab as an independent group, we showed that in the heart as well as in the bone marrow and the liver of humans, we could isolate a population of similar cells that had a mesenchymal stem cell cell surface phenotype. They were clonogenic and showed multipotency. A few years later, from the laboratory of Richard Harvey, it came out a study where they showed that from adult heart, they were able to isolate and grow in culture a population of clonogenic cells that show also multipotency in vitro. And what it was nicely showed that these cells seem to originate from the proepicardial organ. As you all may know, the proepicardium is giving rise via mesenchymal to epithelial transition to the epithelium of the epicardium, which covers the outer surface of the heart. But these cells, by epithelial to mesenchymal transition, can then differentiate to the smooth muscle cells of the heart, the cardiac fibroblasts, and according to some, but this is less shared by the community, also in endothelial cells and possibly in cardiomyocytes. This is nice because when we collaborated with a group of Professor Schneider here in Trieste, Claudio Schneider, and Riken Institute in the framework of the Phantom 5 project, we observed that the transcriptome of cardiac cells with a mesenchymal stromal phenotype was quite unique, distinguishing them from mesenchymal stromal cells derived from the bone marrow or from the adipose tissue. But what was interesting and was in line with the study of Harvey was that the cardiac cells have a mesothelial signature, suggesting a possible link between the mesothelium, epicardium, and this cardiac cell type. The possible in vivo counterpart of mesenchymal stromal cells was first postulated by Paolo Bianco and Giulio Cossu. And I'd like to recall this paper because I remember reading it back in the United States and finding it really interesting, and where they showed that the perivascular cells, primordial periendothelial cells, could in development be set apart and become locally committed mesodermal progenitors. Later on, Paolo Bianco showed also that in the bone marrow, cells with periadventitial features were the founders of the mesenchymal stromal cells of the bone marrow. In more recent years, the work of Sylvia Evans took advantage of genetic labeling studies to verify the in vitro and in vivo potential of cells with periendothelial location. And they showed that cells that were labeled by TBX18 and the transcription factor TBX18 could behave as multipotent cells in vitro, and while in vivo, although they were associated with microvasculature, also in the heart, they were less plastic. Concerning different view, we can also recall that in the ontogeny of the pericytes and perivascular cells is common with that of mesenchymal stromal cells, according to this seminal paper of Slukovin and Kumar, where mesenchymal progenitors that are PDGF receptor beta positive could differentiate in mesenchymal stromal cells and into immature pericytes that then can mature into type 1 pericapillary pericytes and type 2 periarteriolar pericytes, and also have a potential to differentiate into smooth muscle cells. Focusing on cardiac pericytes, several authors have shown that cardiac pericytes can be shown in the human myocardium, that these cells have a periendothelial localization, as they should be, and that they can be identified using antibodies against NG2, CD146, and PDGF receptor beta. However, the same cells stain positive also for mesenchymal stromal cell marker such as CD44, CD90, or CD105. In this case, this cell also showed a multilineage differentiation potential in vivo. Concerning the involvement of pericytes and cardiac pericytes in heart disease, as Frangogiannis recently very nicely summarized, the cardiac pericyte has been largely ignored by investigators interested in myocardial biology in the past. While the pericyte could exert some very important function, for example, in the ischemic heart disease. For example, in myocardial infarction, they could exert pro-inflammatory function during the acute phase via activation, for example, of toll-like receptors and inducing matrix metalloproteinase or release of cytokines, and could also be involved in vasoconstriction and in the no reflow no reflow of the infarcted reperfused heart. In the proliferative phase, the pericyte could, according to some authors, also be responsible for the differentiation towards the fibroblast or myofibroblast fate, while in the maturation phase, it's surely important to stabilize microvessels. Indeed, coronary microvascular disease is emerging as very important in dictating the symptoms and the prognosis of patients affected by heart failure, and also in patients that are affected by macrovascular disease. So macrovascular disease could be also accompanied by capillary rarefaction and reduction of capillary diameter, capillary obstruction, so could be coupled with a reduction of the ability of the coronary tree to deliver oxygen to the tissue. Concerning mechanotransduction and angiogenesis, cells are able to sense a number of mechanical stimuli that range from the ability to spread on cell surfaces, to the ability of sense substrate stiffness, to the ability of sense the level of confluence they reach, as well as the well-known ability to sense shear stress for endothelial cells or the myogenic tone in vessels. How do they sense these physical stimuli? Via different sensors, such as the primary cilium junctions and caveolae that we'll see later on, and that through the modifications of the cytoskeleton and the nucleoskeleton can modulate the expression of genes on target cells. The modulation of expression of genes can be done via modulation, for example, of transcriptional regulators. One of the most studied recently is YAPTAZ co-transcriptional regulator, which could, together with a TID transcription factor, regulate the expression of several genes in a mechanotransduction dependent and independent fashion. YAPTAZ is modulated modulating its intracellular levels via ubiquitination and degradation and nucleocytoplasmic shuttling. Actually, YAPTAs are integrators of a number of different stimuli that are coming from the extracellular matrix, as well as from several transcription factors and G-protein coupled receptors, for example, those that are responsible for the stimulation to respond to the stimulation of the lysophosphatidylate acid present in the serum, and they are important also and have been involved in angiogenesis and also in pericyte morphogenesis. So, moving from this background, we started to think about if we have an alteration of cardiac pericytes in heart failure, and if we could study the mechanotransduction properties of cardiac pericytes and how heart failure is affecting these mechanotransduction properties in human samples. For this purpose, we took advantage of a long-standing collaboration with our cardiac surgery department, and we had the possibility to study both failing explanted hearts obtained from patients suffering from end-stage heart disease of ischemic nature and donated hearts. Of this, we had some very small ventricular biopsies that were employed for histological studies and a small fragment of atria that were employed for the in vitro study. Therefore, a total number of 22 hearts were employed for histological study and 26 hearts for the in vitro studies. First, we studied hearts by histology, and we analyzed the presence of cells with pericyte features, employing some well-assessed markers such as PDGF receptor beta, as well as emerging markers such as CD93 that were suggested by Pearl group as a possible novel marker of pericyte. By counting the number of cells of putative pericytes, this way we showed that with respect to control hearts, hearts of patients affected by ischemic heart disease were characterized by a lower density of PDGF receptor beta positive and CD93 positive pericytes. These pericytes were both of type 2 co-expressing smooth muscle actin and type 1 pericapillary pericytes. We also showed a reduction of CD34 positive and caveolin 1 positive microvascular cells. Caveolin 1 was suggested to be a pericyte markers. However, in our hands, it stained, let's say, half enough both pericytes and endothelial cells. Next, we perform an in vitro study, and we first wanted to see whether the cardiac cells that we cultured in vitro from normal and failing hearts had pericyte characteristics. To do so, we stained these cells for pericyte markers such as PDGF receptor beta or TBX18, and we showed that both pericytes obtained from normal and failing hearts, cultures obtained from normal and failing hearts had a similar fraction of cells expressing these markers. We also stained the cells for the fibroblast associated marker PDGF receptor alpha, and we observed that few of the cells expressed very low levels of this marker. We also observed that half of the cells expressed nestin, which is associated mainly with the type 2 pericytes. Furthermore, the cultures expressed quite homogeneously the cardiac marker HEN2. In order to see whether these cultures had other fibroblastic markers, we also compared the expression of fibroblastic marker TCF21 with cardiac fibroblast cultures, and we observed that cultures obtained from normal and failing hearts were significantly expressing significant less TCF21. Cardiac cells were also able to form three-dimensional vascular networks when plated onto matrigel-coated wells, suggesting that they had a vascular or perivascular phenotype. Finally, we analyzed their transcriptomic characteristic via RNA sequencing. And we compared the RNA sequencing data that we obtained here with those of the paper of Kumar that we introduced before. As you can see here, the cultures obtained from normal and failing hearts have a very similar transcriptional profile, and the closest relative to these cultures are retinal, placental, and brain pericyte cultures. So after this different evidence, we considered ourselves as cardiac pericytes. Next, we decided to verify the differences between the donor-derived and explanted heart-derived pericyte cultures. So the RNA sequencing data were compared, and as you can see here, by principal component analysis, we showed that while the transcriptome of our cultures obtained from normal hearts are quite similar, those obtained from failing hearts are more heterogeneous. Concerning the functional annotation analysis of the differentially expressed genes, we observed that differentially expressed genes were involved in, for example, vascular development, chemotaxis, regulation of filament, of actin filament based processes, etc. And so we decided to investigate more on angiogenesis and mechanotransduction. To focus on angiogenesis, we performed this experiment by employing the supernatant of pericytes derived from both normal and failing hearts to condition the medium of UVEC cells plated on matrigel. And as you can see here, although both pericytes obtained from normal and failing heart support the formation of three-dimensional networks, cells obtained from normal hearts were able to support this network formation more efficiently than those obtained from failing hearts. Subsequently, we decided to study the response of pericytes to a number of physical and biochemical stimuli. First, we evaluated the response of cells to seeding and spreading to the cell plate. And so to do so, we analyzed cells 2 and 4 hours after plating. And we observed that while cells obtained from failing heart respond to this stimulus by increasing in dimension and roundness, cells obtained from from normal hearts respond to this stimulus by elongating and polarizing, a characteristic which is associated also with migratory abilities. With respect to the effect of the fibronectin stimulus on the cells, we observed that similarly to what observed before, pathological cells are mostly larger and more round than normal cells, while cells obtained from normal hearts respond to increased fibronectin concentration by emitting lamellipodia and filipodia and decreasing their circularity, increasing their polarization. Next, we analyzed the response of the cells to physical stimuli such as substrate stiffness. In order to do so, we plated cells on gels or polyacrylamide gels of different stiffness that were coated with fibronectin and on top of glass cover slips. As you can see here in the results, what we see again is that pathological cells respond to stimuli only enlarging, so by hypertrophy, while cells obtained from normal hearts respond by polarizing and by emitting lamellipodia. Another known response of the cell to stiffness stimuli is the proliferation. So stiffness increases the proliferation rate of cells. As you can see here, in both cases, normal and pathologic cells respond to substrate stiffness, increasing their proliferation rate assessed as BRDU incorporation. However, on a gel with a lower stiffness, which is in the order that is observed in muscle tissue, we can see that donor-derived cells proliferate more than cells obtained from failing hearts. Usually, cells respond to substrate stiffness, increasing their intracellular stiffness. And in order to see whether this event was occurring also in our case, we asked to our collaborators to investigate this stiffening property employing an atomic force microscopy approach. Therefore, using the cantilever of the atomic force microscope, cells were tapped, and it was measured the movement of the cell membrane as a function of the force applied to the cantilever. And as you can see here, although cells from normal hearts increase their stiffness as a function of the stiffness of the substrates on which they are plated, cells obtained from failing hearts fail to do so. Since we have seen that different mechanical and biochemical stimuli exert different effects on cells obtained from normal and failing hearts, we started to investigate the sensors that were responsible, that could be possibly responsible for this response. First of all, we analyzed focal adhesions, and by immunofluorescence, we stained focal adhesions and the whole body of the cells, and we evaluated the volume, the area fraction of the cell that was covered by focal adhesions. And as you can see here, independently from the concentration of the fibronectin on the cell surface, cells obtained from normal hearts have more focal adhesions than cells obtained from failing hearts. This is paralleled by the activating phosphorylation of focal adhesion kinase, which is higher in cells obtained from normal hearts with respect to cells obtained from failing hearts. Since it was also described that matrix rigidity is transduced in cells via activation of P130 Cas, we also analyzed whether this pathway was similar or differential in pericytes obtained from the two cell types. And as you can see here, although pericytes obtained from failing hearts have more Cas, the phosphorylation of Cas has a trend which does not reach significance, but has a trend to be less active in cells obtained from failing hearts. Another possible mechanosensitive pathway is mediated by caveolins. And in order to see whether this pathway was differentially regulated in cells obtained from normal and pathologic hearts, we stained both the cell types with an antibody against caveolin 1 that, as we have seen previously, was suggested also to be expressed by pericytes. As we can see here, caveolin 1 is more expressed in cells obtained from normal hearts, while cells obtained from failing hearts do express less levels of reduced levels of caveolin 1, and this is also supported by real-time PCR experiments. Caveolin transduces its activation also it could modulate the raw small GTPase that is important for actin remodeling. And as you can see here, we performed an active raw pull-down experiment, and we observed that although there is a trend toward a reduced level of raw activation in cells obtained from failing hearts, this is almost significant, but does not reach significance. Which is instead, for sure, significance are the differences in the actomyosin cytoskeleton of these two cell types. And both actin and stress fiber thickness and the number of stress fibers per area covered by the cell, and also the non-muscle myosin are significantly more abundant in donor-derived pericytes with respect to pericytes obtained from failing hearts. Finally, mechanical stimuli are transduced into a biochemical response and gene expression response via mechanosensitive transcription factors. Among these, YAP, as we previously anticipated, is emerging as a very relevant co-transcriptional regulator, and another is myocardium-related transcription factor A. In this experiment, we plated cells obtained from normal and failing hearts on top of gels of lower and higher stiffness in the presence of lower and higher concentrations of fibronectin. As you can see here, cells obtained from normal hearts show a significant increase in the nucleocytoplasmic ratio of YAP as a function of the substrate stiffness, while cells obtained from failing hearts do not respond in a similar way. Myocardium-related transcription factor A has a similar trend where its nucleocytoplasmic ratio is increased as a function of substrate stiffness in normal hearts and is not occurring in failing heart-derived cells. Furthermore, YAP is also modulated as a response of the fibronectin levels in the cell substrates only in cells obtained from normal hearts, while this effect is not occurring in the case of cells obtained from failing hearts. While myocardium-related transcription factor A does not seem to be so responsive to fibronectin concentration. Concerning YAP the YAP levels, we also analyzed the levels of total YAP and the phosphorylatory events of YAP that could regulate in a hypo-YAP dependent pathway this co-transcriptional regulator. And as you can see here, we did observe significantly higher levels of total YAP in cells obtained from normal hearts, but this was in spite of not finding differences in the phosphorylation of status of YAP. However, the total levels of YAP can be regulated by via the ubiquitin proteasome system by the E3 ubiquitin ligase beta TRCP. And consistently, we observed that in failing hearts, we showed significantly higher levels of beta TRCP in cells obtained from failing hearts. Consistently with the immunofluorescence and Western blot data, we observed that 2 out of 3 of the 3 target genes that are regulated in a YAP TED1 only dependent fashion were significantly more expressed in cells, in pericytes obtained from normal hearts with respect to pericytes obtained from failing hearts. This is also an in vivo counterpart. In fact, when we went back to the tissue of patients and controls, and we did the immunostainings on this to see the YAP nuclear translocation in pericytes or microvascular cells of controls and ischemic heart disease tissues, we observed that YAP has a nuclear localization in a significantly higher fraction of PDGF receptor beta positive pericytes in controls with respect to failing tissues. And the same is true for caveolin 1. At a transcriptional level, hearts obtained from controls show higher level of expression of the 3 YAP only regulated genes, and 2 genes that are CTGF and CYR61 that are co-regulated by YAP and myocardium-related transcription factor A. Another level of regulation of YAP comes from G-protein coupled receptors. Among these, it has been shown that serum components, and especially lysophosphatidic acid, may regulate the nucleocytoplasmic shuttling of YAP, and is also involved in controlling actin dynamics. As you can see here, if we compare the expression of lysophosphatidic acid receptor in pericytes obtained from normal and failing hearts, we observe that actually cells obtained from failing hearts express significantly lower levels of lysophosphatidic acid receptor 3. Concerning the ability of the serum to modulate the actin cytoskeleton, indeed, if we deprive cells from serum, as expected, we see a downregulation of F-actin. And if we evaluate the YAP translocation as a response to serum deprivation, as you can see here, we have a significant down regulation of the nucleocytoplasmic shuttling in both normal cells and failing cells in serum-free medium. Similarly, myocardium-related transcription factors shows a similar trend. This effect is also observed when we have very high levels of fibronectin in the cell substrates. However, in this case, at the same level of at high levels of fibronectin, the levels of nuclear YAP is higher in controls. The activation of YAP via mechanotransduction has been shown to be to involve ROROC signaling. Therefore, we analyzed the sensitivity of YAP to a ROC inhibitor, Y27632. And as you can see here, normal cells cultured in low levels of fibronectin stimulation show a downregulation of the nucleocytoplasmic shuttling of YAP, while cells obtained from pathologic hearts do not seem to behave to reduce the nucleocytoplasmic shuttling of YAP as a response to ROC inhibition. This behavior is similar to what has been observed in caveolin 1 null cells. However, the ROC inhibition effect was somehow reduced when cells were cultured at high fibronectin concentration. A similar outcome was observed when we used actin depolymerization drugs such as latrunculin A, where cells obtained from donor hearts are sensitive, cells obtained from failing hearts are less sensitive, and cells obtained from normal hearts also in this case are sensitive also independently from the fibronectin concentration. Last, we analyzed the activation status of MEC-ERK signaling. We focused on this for different reasons. One is because in the functional analysis of the differentially expressed genes, we observed that positive regulation of MAP kinase cascade, regulation of ERK1 and ERK2 cascade were differentially expressed by the two MEC could modulate and positively regulate the expression of beta TRCP. Therefore, we compared the levels of ERK in normal and pathological cells, and we observed that pericytes obtained from failing hearts showed an increased level of ERK and phospho-ERK that could be reduced with a MEC inhibitor. Importantly, when we employed this MEC inhibitor in cultures obtained from normal and failing hearts, we could show that we could restore the nuclear localization of YAP independently from the fibronectin concentration in the substrate and independently from the pathological status. And this was associated again with an increase in the total levels of YAP that were paralleled by a trend to reduce beta TRCP levels. To summarize, what we showed here was that explanted failing hearts are characterized by microvascular rarefaction and a reduction in the frequency of pericytes, and that these pericytes are characterized by reduced nucleocytoplasmic shuttling of YAP in vivo. We also showed that we can obtain and culture in vitro cells that have a pericyte immunophenotype, a transcriptional status, and a transcriptome. These pericytes have reduced proangiogenic features when obtained from failing hearts, and the pericytes obtained from failing hearts are less capable of responding to mechanical and biochemical stimuli. This is due to a complex series of alterations that we describe in the pericytes obtained from failing hearts. Specifically, we show a reduced activation of focal adhesion kinase, a trend to reduce the activation of CAS, and for sure we show differences in the actin cytoskeleton of these cells. Furthermore, we show a reduced expression of G-protein coupled receptors such as lysophosphatidic acid, coupled with a trend towards reduced activation of Rho signaling, reduced levels of myosin 2 and actomyosin, and a reduced ability to transduce extracellular rigidity into cytoskeletal tension. We also showed defects in caveolin-1, which has been shown to be important in transducing YAP signaling in an F-actin-dependent fashion. And indeed, as anticipated, we show reduced levels of F-actin. Caveolin-1 defects are also coupled with hyperactivated status of MEK-ERK signaling. This is also consistent with literature data where caveolin-1 null mice show cardiac hypertrophy, which is associated with hyperactivation of MAP kinase in cardiac fibroblasts. Furthermore, we show that, consistent with other data that have been obtained in tumors, that ERK activation is associated with increased levels of beta TRCP, that in our case are also coupled with reduced YAP levels. Last, we show that by inhibiting MEK-ERK pathway, we can interfere with the beta TRCP pathway, and most importantly, we can increase the nuclear localization of YAP. This is my last slide, and I want to thank all of you for your attention. I want to thank all of my group, especially Daniela Ceselli, which is sharing with me the responsibility of our research. Irene Rolle, she is a great molecular biologist, and she has done a lot of the molecular work here. Our collaborators from the cardiosurgery department and cardiology department, and and our collaborators from CNR who did the nano indentation study. Last but not least, Elisa Voglio, which happened to be one of my previous students and now is a brilliant researcher in Bristol Heart Institute. And I thank you for your attention, and now I'm happy to take any question.
Great, thank you very much. It was really fantastic, very exciting. Please, everybody, continue to enter any questions you have in the Q&A window. There's a few of them that have popped up so far, so we can start a little bit of discussion here. Lucio Barile, maybe, says, according to your data, pericytes proliferate in controlled conditions. Is this phenomenon accompanied by apoptotic processes that guarantee a balance in vivo?
Sorry that I was trying to read it. Okay.
Okay.
So the question is whether the proliferation is balanced by apoptosis. So we actually studied the proliferation effect in a very artificial system, so in vitro, following a very high stimulus, which is a very rigid surface, which is not the same, of course, as the heart. If you remember, actually, when the cells were plated onto softer matrices that were on the elastic modulus around 16 kilopascal, actually the proliferation rate of the cells was much lower. So the answer is probably in vivo they behave very, differently. What we wanted to stress out is that their ability to respond to stimuli is quite different. Even in these softer substrates that are more closely mimic the in vivo situation, actually cells from normal hearts proliferate more than cells from failing hearts. Whether this is balanced by apoptosis is a good question. We could follow up in interrogating our cells and see what happens.
Mm-hmm. Kind of along those lines, I was actually wondering if you anticipated or if you've seen differences between pericytes in different regions of the heart, that either in different regions of the heart, or would you anticipate differences in early heart failure, say, versus late heart failure?
Okay. Different regions of the heart we did not investigate so far for one simple reason, because of the donated hearts. We need to use what is available, and usually what's available is a very small piece of the atria that has not been implanted in the recipient. And so we have tested mainly this cell type. For sure, I'm pretty sure that if you start to focus more on differences, and you start to culture and compare cells from different regions, you will end up with for sure differences that also probably are also dependent on the different also embryological origins. Since, for example, the atria come from the secondary heart field, the left ventricle from the first heart field. For sure, is something different. Could be anticipated. Let's say.
Sure. Giancarlo Forte says, thank you for the nice presentation. YAP is generally found to be activated in pathological hearts in cardiac fibroblasts and myocytes. Can you speculate why pericytes might be different here?
Actually, yes, there are. I mean, I've seen different reports with different outcomes, meaning that in some works actually YAP has been shown to be less translocated in failing hearts. In others, they say that is more. Probably this also reflects the status of the cardiac evolution and the compensation evolution. These are end-stage failing hearts. I can anticipate that in this case we see that YAP is absolutely less translocated in cardiac myocytes of this patient, but is like 100 to 0. So in this case, myocytes parallel the pericyte. But whether this is one extreme condition like the exhaustion of a compensatory phase may be interesting to be investigated. If I remember correctly, there was a paper by Professor Martin who investigated YAP signaling and showed that by interfering with YAP signaling, it could promote somehow regeneration, but then it could lead to the opposite, if I'm not wrong. Now, like to a hypertrophic cardiomyopathy kind of phenotype. So maybe there could be something similar going on.
Sure. Okay, John Lederer wanted to know a little bit more about the origin of the cultured pericytes. Do you think these are arterial, capillary, venule pericytes?
Yeah. For what we have seen, but only based on a few markers, we have seen that these are a mixture of capillary and arteriolar pericytes, because for example, NG2 and also nestin are not expressed in a very homogeneous fashion, but in that case, especially nestin, you have cells that are very positive and cells that are almost negative, while, for example, TBX18 or other markers are pretty homogeneous in their expression. So my idea is that these are mixed cultures. Indeed, maybe I am anticipating something. We wanted also to investigate this at a single cell RNA sequencing level, but then COVID came out and we stopped our our study. So well, this I think
gives you more to do.
Yeah, it could be very interesting to see in these stromal cultures the different subsets, how the pathology may influence these different subsets.
Yeah. Giuseppe Gianfrancesi.
Oh,
sorry. Says, thank you, Dr. Bertrami, congratulations. This is a very comprehensive presentation of some excellent work. Do you already have some ideas for future developments of this study and your next steps?
Yeah, I mean, there are things that are going on in other labs that, but I don't want to anticipate too much.
That's the next grant application, right?
That are very promising, but are other labs. And actually, as I anticipated, our idea was also to try to see if we can understand the complexity of this stromal cell phenotype, to see whether we have different subsets that are maybe plastic, and that so that a cell could transit from one cell type to another cell type. For example, the paper that I was mentioning by on TBX18 lineage tracing, you know, they neglect that pericyte could could transdifferentiate into myofibroblasts or fibroblasts. But there are other papers coming out that they say that there is a more ductile phenotype. So I would be very intrigued to see whether this is occurring, whether these cells are more plastic, also taking into account their ontogeny, as we mentioned before.
Right.
But
Well,
sorry, Stephanie, do you mind if I just jump in here?
oh, yeah, please.
One of the things, thank you. One of the things that I'm actually beginning to question is when we culture cells in these 2D conditions, and yes, they can change into God knows what else. But I don't know if you're familiar with Cesare Terracciano's work on fibroblasts on cultured slices, cardiac slices. The behavior of these cells is very different all of a sudden. So I do wonder whether we're making too many assumptions by looking at simply culture conditions on 2D, glass or plastic or fibronectin, whatever it is, that may actually differ from in vivo environments completely.
But this is, of course, it's very true, and it was the first thing I pointed out in the first answer. So we are testing a cell in an extreme condition. However, we should not neglect that we can learn a lot from this, because, for example, by comparing the transcriptome of these two extreme types in vitro, then we started to understand, for example, as anticipated by Stephanie before, the autophagy inflammasome study, and we were able then to go back to the tissue in, for example, in dilated cardiomyopathy patients, and to see whether the same alteration that we observed in vitro could be traced back in vivo. So I think that this back and forth between the cell culture, go back to the tissue, come back to the cell culture, could add something. Of course, it would be better to have a better in vitro system, but we discussed just yesterday with a bioengineer. Of course, for example, engineered heart tissue, you increase the complexity of the tissue, but then it's very difficult to study it, so you have to go back to histology. So you cannot interfere that much with the system. So it's a balance between different needs, let's say.
No, I fully agree.
Well, and the way that you and others are looking at different types of stiffnesses and substrates, you're really assessing the mechanoresponsiveness of cells, which is something that we can apply in vivo. So it's getting better.
One question, because we're trying the same thing actually here. We're trying to culture cells in different substrate stiffnesses in a very similar range to what you've been showing. We're buying these plates from
IBD.
There's a couple of them that I can't remember right now. Commercial suppliers. But you've used these gels, right? You've used just acrylamide gels. So did you measure the substrate, or did you just follow that recipe? I remember there was a paper that came out with particular recipe.
We have followed mainly the recipes, but then we had also the possibility to have a collaboration also with nano technologists who tested the stiffness on cells on this, and so on. So we are quite confident of that. With the gels, it's tricky. It was not very easy. I have to say that this was not an easy paper to be delivered because there are two PhD students who finished their PhD that I should acknowledge, that are Elisa Mazzega and Andrea Zanello, who spent their PhD, and I was telling them we must see differences between the two cell types, but they were struggling because the cells didn't want to attach to the substrate. So Elisa in this was very very smart, and so she she started to follow other recipes. She photoactivated the gel before putting the fibronectin on top, and this was really making the difference, because on very soft substrates the cells do not like to adhere that much. So yeah, it's not that easy.
It's okay. It's normal for PhD students to hate a supervisor. It's okay. Sorry, I'll shut up now.
Oh, okay. A couple of quick questions to get through here. Aram Babakar, thank you for the excellent talk again. How do you think pericytes are contributing to the systolic and the diastolic dysfunction of failing hearts? Do you have any thoughts on how that might be directly happening, or how they're contributing to that injury, and if you have plans to study that?
It's a very good question, but so far, I mean, we did not evaluate in this specific paper, for example, the secretome of these cell types. Of course, by modulating their biological response, the cells also modulate the secreted factors, and so modulate also the kind of matrix that they deposit. So one idea would be to do a biophysical study on the matrix that these two cell type deposit, but this is something that has to be done. So if somebody wants to collaborate with this, we are really happy to share our things.
And the last question that was asked, it kind of follows along that. Do you think that there's a specific aspect of heart failure that might be pushing the pericytes toward a particular phenotype, or maybe that's getting at, you know, what their job is, I mean, in relation to capillary systems, but yeah, is there anything that stands out to you, I guess?
Okay, we did not, I mean, we focused on in this case, on ischemic heart disease. But what we can tell is that we have a dysfunction of this cell type also in dilated cardiomyopathy, for example. And also you can induce senescence and dysfunction of the cells also by hyperglycemia. So actually the correct answer is I don't know the impact of each and different stimuli to this aspect, and also this could be interesting to be investigated for sure.
Yeah.
Okay.
I think, one last one. Do you have plans to look at transmembrane mechanical ion channels in pericytes?
We did. I mean, we did electrophysiology studies back in 2007, and we should. But these were mainly focused on studying the maturation of the cells towards kind of a neuronal-like phenotype. What we observed is that when you induce a kind of this phenotype, you can see some sodium currents that do not reach, of course, threshold and action potential. And you can see also potassium currents like delayed rectifier type potassium channels. But then we did not look specifically to ion currents. Although if I remember correctly, among the differences between the normal and pathological phenotype, there are potassium channels, if I remember correctly, that puzzle me, which may be also interesting to follow up in future studies.
Yeah. Cool. Okay, well, I think if nobody has other questions, I mean, we can keep chatting for all evening, but there's no other pressing questions from people.
Antonio, you've done very well. I mean, you know, you've answered a barrage of questions and carried 70% of the audience with you into
Right.
an hour. So, you know, I think that was quite impressive. So well done.
Thank you very much. Now I need some water to rehydrate myself.
You know, I mean, first of all, I'd like to actually thank both of you, Antonio, for a fantastic presentation. Really, really good handling of the questions. I think, you know, you've gone way beyond the paper as well, which I think is quite nice because these webinars allow that.
You make me blush, so
it's deserved. And Stephanie, thank you for a really great sharing. I certainly couldn't chair this session. This was definitely not my area of expertise. so thank you both for the time, and thank you for opening of the ISHR JMCC webinar series for this year. So it's fantastic.
Thank you so much. It was really a pleasure and an honor.
No,
yes. Thank you. I mean,
always fun to talk about science.
So I mean, obviously you may have some closing statements, but I would just like to remind the audience the next webinar will be the paper of the month as selected by the editors of JMCC, which is basically paper of the March edition. And that's, I believe, on the 28th of April, actually, is the next paper of the month webinar. So yeah, I'd like to thank you both, and of course, you may have some closing remarks, but well done.
I just want to thank everybody, and again, JMCC and the International Society for Heart Research. Thank you again. And it was really a pleasure to meet you both, Davor and Stephanie.
Yes, it was very nice to meet you both too.
Likewise. I hope we meet in person soon as well.
Yeah, hopefully.
Yeah.
Okay.
Thank you very much, and enjoy afternoon, evening, you know, morning, whatever it is for you.
Yeah.
Okay.
Thanks, everybody. Take care.
Thank you.
Bye-bye. Bye.